today, I'm going to talk about respiratory rhythmogenesis and genetic architecture. And the goals are, this is where it starts, is to define breathing as a complex trait, explain how rodent models can be used to dissect gene actions, and describe the concept of a genetic map. Now, a genetic map is shown right there, right? You got lots of genes down here, and you got lots of traits up here. So what's in between? Well, you got proteins, you've got cells, you've got systems, you've got physiology, you've got X, Y that makes this trait, right? Tidal volume and frequency make ventilation, sleep, and I don't know, breathing makes sleep apnea. All these things do it. And so it's interesting to sort of say, well, what are the genetics of all these things? And I'm going to sort of take you through mostly adult, but I'm going to show you some other things as well. And these are the Grinnell relevant. So here you go. You've got recurrent apneas and sleep apnea and sleep disordered breathing. Should I get this out of the way? How do I get this out? Hit it? Yell at it? What? Can't do that.
More.
More? Here?
Down 2 more.
Here?
Yep.
That worked. Okay. Let's see, is there another hide in there? Don't know. Okay, anyway, what you see here is that a complex disease is the result of common mutations. Genes are neither necessary nor sufficient, but collectively with a different percent influence interact to tip the balance towards diseases of ventilatory control like OSA. So we always think of body mass, sleep, ventilatory drive, ethnicity, but then there'll be genes that are not genes that you suspect, which increase or decrease your susceptibility. And so it's not just a positive gene, it's also a negative gene. Ventilatory drive has lots of genes to get up to tidal volume and frequency. Body mass, head form, have lots of genes that go up there. Sleep and sleepiness have lots of genes that go up and give you sleep and sleepiness. And then gender and ethnicity has lots of genes, right? And so what I'm going to be talking about is a whole mess of genes. You're not going to memorize these things. You're just going to know what the concepts are. So the different approaches that are on there, Shork and Lander in 1996, and Nick Shork came here and talked about blood pressure. And that's what got me going on genes and breathing. And he gave this talk and he had this approach. Now, who's Lander? Who's Eric Lander? Well, he was the head of the genetics at Mass General. He got in trouble through various other things, but he was one of the big drivers of human genetics. And then there are other people that were in animal genetics, and they all kind of came from kind of like a small little workshop of geneticists in the 80s and 90s that were looking at disease. So twin studies usually in the family, population studies, and animal studies. So we're going to go through all of these particular ways to try to dissect complex disease. Human subjects, twin studies. So twin inheritance is about 20%. This is the most recent largest group of twins, 72 pairs. Both twins had an AHI greater than 15, 16 pairs. If you call 15 being disease and AHI, you'll see that. But, you sort of say, well, 40% had an AHI greater than 15, and this included both monozygote and dizygotic twins. So that if you had a shared genetic background at a twin level, you had a chance of having sleep apnea that was higher than random. This is the heritability of finding another member of with a high AHI. And this was Cleveland Family Study, 1996. This was done here with HSTs out in the community, doing the entire family, about 180 individuals. But the odds ratio, if you had one person in the family of finding two, was 2.4. If you had one, it was 1.5, 2.4, but it's stepwise. That is, the more people you had in the family, more likely you were going to find that. On the other hand, it was really not enough to be able to sit there and do this. And this is breaking it out a little bit more. If you had greater than 5 in a family, this is 1, this is 2, and this is greater than 2. So greater than 5 is fairly common, which is sort of like what we think. But if it's greater than 15, 15, 51%, 33%, 16. So it said that the gradations, though, also run in families. It was not enough to test everyone in a family for disease, and that was the modeling that was there. It's not as though if you find a couple people in there, you should test everybody to see if they have it, but it raises your awareness, and it's the foundation of why we ask family history. So population studies, affected versus unaffected, qualitative trait linkage. So you have a disease, you're interested in trying to find out something about it. And you have two things. You got hereditary or you have complex. Hereditary is what is really what Eric talked about. 2020 of all the inherited pediatric sleep genetic syndromes.
I remember the pediatric focus one, yes.
Yeah, so you got CCHS, FOX2B, Rett, Pierre-Robin, Marfan's, myotonic dystrophy, Treacher-Collins, Prader-Willi, trisomy 21, Hunter's achondroplasia, all those sorts of stuff. And you can recognize that right in the first kind of 2 years of life. So they're not completely monogenic, but they're really evident that there's something wrong. And then they went on, you can find those genes. What we're going to be talking about today is here, the complex. And the common theme is you do a gene-wide association study. And now you can do this relatively cheaply to find out what polymorphisms are along the entire 23andMe. And you can do all that sort of stuff. And you can do it relatively cheaply, cost maybe $8, $9. That's why they can sell you this for 90 or whatever it is. And then you want to find out what that is. You want to look at a population, you want to replicate it, and you want to see if they're disease susceptible. And there's all the genes, right? All the genes related to those things. So this is a pretty good slide to think of it. There are very rare genes, they're very common genes. Very rare genes are the ones that, like FOX2B, that cause the congenital. There are rare alleles causing Mendelian disease. They probably have some other rare variants to detect very hard. But if you're looking for those sorts of things, you find these rare genes that pop out because you see them in a pediatric population. But the other is these few examples of high effect common variants. There are very few. One of them might be the FOX2B gene that's in the parents of the kids that have FOX2B that show that they breathe funny. But most of them are common variants that are implicated in common disease. And what that means is that you have lots of these polymorphisms that contribute in aggregate to the determination or the opportunity to have the disease. Doesn't mean you have it, it just means that you have the opportunity for disease. So this is the first one that I knew about, 2011. This is a whole genome scan for 24-hour respiratory rate of major locus. And this is a locus when you didn't do genes, but you did parts of genes. You could break them up into parts pretty easily. And you could find that the evidence of linkage found at chromosomal those 4 locations. The strongest evidence of linkage was found for respiration rate during sleep, with LOD scores, which are modest, of 2.36. Those are very modest. Could be attributed to a quantitative trait locus near this marker. Don't know what it is. No one's really pursued it very much. But I think this was really a kind of an interesting thing that despite all the ways of trying to fight those genes to a trait, if the trait is breathing, and then the trait is breathing during sleep, you can get some sort of genetic marker that associates with that after you adjust for all the multiple associations that go on with the genes. And so it's sort of, to me, it was like, wow. But the best part of this was the beginning of the first sentence. Billions of mammals, including humans, depend on rhythmic breathing to regulate one of the foremost aspects of homeostasis, the appropriate exchange of oxygen and carbon dioxide. So finding genes is a worthy cause, right? And so it's really evolutionarily important. So what about sleep apnea? Some progress. And I'll show you really what came out and some of the things that were talked about at ATS that have just come out. This is a previous one that had a meta-analysis of 3 different genetic GWASs that were small. They pooled them together and found out what it is, TNF alpha. OSA was the only one that came out. All the rest, crossed 0. As you can see, some of these studies got excited because here it goes on all 2007, ApoE4, and he was going, oh, look at that, it's Alzheimer's. But then 2 others didn't replicate, right? You got to replicate these things before you start doing this. And ApoE2, E2, ACE. ACE has held up pretty well, interestingly. So recent GWAS SNP studies, the Million Veteran Project that you see advertised over at the VA, the FinGen and MGB, which is a US based, doesn't have as many, but the Million Veteran has over 560,000 individuals. The total of this whole group is almost 1 million individuals. And so this is the one for the Million Veteran that was published about the end of December. They had GWAS of this many people, stratified by biological sex and harmonized for race, ethnicity, genetic ancestry, which is HARE, white, black, Hispanic, and Asian. This is what you need. You need these huge groups. Consider both the BMI adjusted and unadjusted models. We replicated associations in independent data sets, analyze the heterogeneity of OSA genetic associations and sex groups. This is where I'm pleased that I'm an MD. I take care of the patients. I provide the samples. So I don't have to do the statistics, right? I can hire somebody to do the statistics. That's the best part. Although the statistic people take credit. They go, I found the gene. You say, well, you didn't do it unless you had 900 whatever it was, 500 people drawing blood across the US. Findings of the MVP were mostly male, as you expect from the VA. OSA prevalence was about 21%. And this is how you read it. These are called Manhattan plots. And this is a Manhattan plot in which unadjusted for BMI, and this is a Manhattan plot, just the reverse of this one. That's adjusted for BMI. So in this one, it says, oh, look at all these things, and greater than 6 is okay, greater than 10 is terrific, greater than 15 is wow. And here you've got this huge signal here, you get excited, and it's nothing's there after you adjust for BMI. So that's a BMI level. It's not an OH, not a OSA. Usually it's AHI. Nothing else. They didn't do sleepiness. They did, adjusted for sex and things of that sort, but they didn't do a lot of stuff. And here it is below, adjusted for BMI, and there are a few of them here. And they reach a little bit of significance here, which is at a point here, the same point here, about 8. So it's over 6, which is everybody gets excited about, but it's really not as high as this one, 40 and things of that sort. But they didn't stop there. They also were able to pull in all the data that came from the FinGen, MGP, and here's on top is the MVP, the Million Veterans Project, Manhattan plot. And here's the one where they put all of them together. And what you see here is sort of like, this thing is still there, so that's good. They line up pretty well, but some of these things start to jump out. And that's because the more you have, the more opportunities you have, the more heterogeneity you have, the more you're able to sort of say, is there anything there? And I point out these because these are ones, the red ones there are ones that were there after you adjusted for BMI. And that's a really cute little thing. That is, if you take away the BMI and certain genes pop up and get to be important. Because we got half the people with sleep apnea don't have obesity. So that's kind of interesting. And then there are two others. So everybody says, the next one says, well, what are they? Scaper down here on the right is S-phase cyclin A associated protein residing in the endoplasmic reticulum. That links directly to sleep apnea, doesn't it? Come on, man. What's metal 1,5? It's a mitochondrial gene. Oh, that may make sense. Another one is SYNGD. It's a syntrophin gamma 1 protein coding. It includes idiopathic scoliosis and scoliosis itself. Sleep apnea, it's really, boom, that gene, Mary. So you see actin 1, that's good. Guanidine nucleotide exchange factor essential for activation of RABA5A during engulfment of apoptotic cells. What? Mars2, methionine sulfoxide reductase. And DLEUI in molecular biology deleted in lymphocytic leukemia 1, also known as something else. It's a long non-coding RNA. So we're going to find a gene for OSA this way, right? Yeah, it might, but you're going to need more and more people. Then you're going to need to figure out what you replicate, and not just in these three, but other three. And then you have to figure out other things. Now, there's some interesting things that happen about this. So they found 18 and 6 genome-wide significant loci for BMI adjusted, unadjusted, and BMI adjusted. 17 were previously reported. Sex differences were there and widespread, likely associated with multiple risk factors. OSA shares genetic underpinnings with several phenotypes, shared etiology and causal pathways. What it is, it's pleomorphic. That is, it can cause chronic lymphocytic leukemia, but it can also cause OSA. So that's how genes work. This was more interesting. It came out and it took the genes in OSA and took the genes in the OSA, whether or not they had severe COVID or not, and found that there's a causal link to COVID with OSA. Causality is driven by the BMI, which is what everyone suspected. And the BMI independent association was seen with earlier supported SLIC4 and a MECOM locus. These are solute carrier 9s and ectopic virus integration sites. All right? Now, this is not something you learn in medical school. This is not something you learn really in biology. This is something you learn in genetics, and you're trying to learn this sort of stuff, try to figure out. But the technique is important to understand. And so, in summary, multiple pathways likely determine the risk, and a better approach. So you're going to find gene variants for obesity, for craniofacial structure, for neural control, for other things, whatever you can think, hypoxic load, sympathetic load, arousal load. Those are the three that were really talked about at the ATS. And the better approach is to study gene variants to intermediate traits. No one really talked about genes to the causal pathways. I think somebody must be doing it. But if they were able to get causal genes for p-crit and be able to dissect out that's still a complex trait. One for gain, one for those sorts of things might be interesting. Okay, so why did I get excited about this? Well, I got excited because Short showed me how in animal studies, you can dissect out genes that relate to blood pressure variations among different strains, and that you could take those genes and you could map them back onto to humans and understand more about. No, you say, wait, a rat or a mouse gene to a human gene? Guess what? We're all mammals. We got genes, we got stuff. And if it's not exactly that gene, at least you know that that pathway might be important. And so that's what I started doing at that particular time. Animal models are free of confounding effects of disease and length of illness inherent in human studies. I don't know if a person's had sleep apnea for 20 years, then they had sleep apnea for 10 years, 5 years, or 2 years. I don't think anybody has 2 years. Efficiency of gene finding interactions for a relevant phenotype or intermediate trait. And then you can look at studies of function in vitro and in vivo. So these are the naturally occurring OSA, and they're not very helpful. That is, a breath hold model with the elephant seal and the craniofacial is not going to be very helpful. And yeah, there is a gene, GWAS for elephant seals that's been published. Yeah, there is one for bulldogs that's been published. And they do have a sleep apnea, but these are really hard to study in the lab, especially if you want to sit there and say, I want to take that gene and figure it out. So what we looked at and started looking at is acute hypoxic responses. And what that is there's a baseline, you give them hypoxia. First minute or so, they get to a peak of ventilation. Over 5 minutes, it rolls off. And then after you stop it and you give them 100% oxygen, you have two choices. One's called post-hypoxic decline. That is, you go below the value of baseline, or short-term potentiation. A lot of people like the short-term potentiation. They talk about it a lot. They say that that's really there in hypertension more than anything else. But this also happens, and it happens in animals. It's been studied. 2001. So we started out with brown Norway and Sprague-Dawley rats. Brown Norway is a steadily colonized Europe and England in the early 18th century. Brown rat may be able to thrive in environments that were not suitable for black rats. How about that? And it was inbred. This is inbred. Brown Norway is inbred. They're inbred by a company. Sprague-Dawleys are outbred. They're albino mutant, identified in the 19th century, has been bred for research for 7 years. They're pretty hardy. A lot of stuff comes out with them in pharmacology, a lot of CNS stuff. We tested them by putting them unanesthetized into a box. This is really a clear plexiglass box. You monitor the temperature, humidity, oxygen, CO2. Put a gas mixture in there. You create a suction through the box to clear the air, but you also put a pressure transducer in which the amplitude of that pressure transducer tells you something about tidal volume and frequency. Is it perfect? No. Is it what you could do if you had a thing on the face there? No, it's not. But it's close enough, and it's about plus or minus 10%. So what you want to do is figure out whether or not something goes a little bit more over those things. And I have the papers. There are about 8 papers that look at this box, and 8 of them were by biomedical engineers trying to make it better and complain about this, that, and the other, which made the values for tidal volume go up or down. But frequency is pretty good. You can watch the frequency. You can say, yeah, it's the frequency 120, okay. And what does it say? 121. Okay, that's okay. And you can see if they move around, it just is a terrible signal. So this is what the Sprague-Dawley looks like on top, and the brown Norway on the bottom. Sprague-Dawley at this particular thing. Maybe this brown Norway doesn't breathe as much. These are not easily harmonized for each strain. Everybody complains about that, but look at the difference. One kind of goes crazy with hypoxia. One just sort of sits there and goes, what was this? 10% oxygen. And this was the 5th minute. So what's interesting is the first minute goes up a lot in the Sprague Dawley and comes down, and the brown Norway goes up a little bit and sort of like, So what did we found? Well, change in minute ventilation with CO2 and hypoxia, Sprague Dawley and brown Norway. Sprague Dawley had 120% increase in ventilation with 8% CO2, while the brown Norway did 2%. I actually did a really good study. We made a real hypoventilation rat. That brown Norway is a hypoventilator. And then in terms of the same sort of difference, it's a lower ventilatory thing. Those little Sprague Dawley and brown Norway. Now the Sprague Dawleys are not inbred, but those are big differences. So you take those two and you cross them to an F1, and then you cross them again to the second generation, and you mix up all the genes, right? That's what happens. Mix up all the genes. And you study each one of those individual F2s. You chop up each one of their individual genomes, and you say, okay, to that individual animal, what links to what value? Whatever you want of the trait. And you kind of pile those up, and you end up with a linkage, an association between a part of a gene to a trait that you measure. So frequency and tidal volume, and these were the linkages that you could find to all sorts of things in the genome. And we took genes that were normally distributed, et cetera, and the genotypes were there. You correlate the genotypes on each side of the gene itself. It could be a brown Norway, it could be a Sprague Dawley, or it could be a brown Norway, Sprague Dawley on each allele. So this is then chromosome 7 for minute 1 of hypoxia in both genders, in which it didn't really get very high at all. But to our surprise, if you added those two genders together, it popped up there above 6, which was pretty impressive for both genders in these markers. Now, we weren't doing individual genes at that time. Doing this, the individual genes you had to work a little harder at, but somewhere in here in chromosome 7, which is underneath here, can be anywhere between 10 and 30 genes in here. There was some region in which it really correlated pretty well. So this is proof of concept that you could find this sort of thing. A second observation I want you to realize here. Let's see, time is that this was a frequency of time. So you took the entire population and you looked at minute 1, and then let's go to minute 5. Minute 1 had the highest association, the breathing level at minute 1. So remember, between those 5 minutes, the breathing would go down. Now it goes down because CO2 levels start to fall, all sorts of things happen. But you can sort of see that different linkage in minute 3, minute 2 to minute 1 to minute 4.5 to 5, that this is a pretty dynamic thing, that this animal's genome is making something that it's attentive to in the first minute of responding to hypoxia. It's dynamic. Now, the gene people hated me for that because they said, oh, we want steady state physiology that we can link to them. And I showed them this and they went, oh, well, we never thought of looking at that way. Yeah. So let's look at what else we. Here's another one. So I told you chromosome 7. And chromosome 3, though, D3R12 is another QTL. And you can see that in frequency itself, brown Norway is low if it had that particular brown Norway chromosome 7, but it was high if you had the chromosome 3. And it was the opposite here. So what it is that this again showed what Short showed in blood pressure. There's some genes that make it go up. And then there are some genes that make it go down. So it's really, how does that, how does that play out? And that's sort of like blood pressure, right? It's sort of like BMI. Some genes that go up, some genes that go down. Some people eat a lot and don't gain any weight, and some eat maybe modest amounts and gain weight. I mean, that's the way it is. So the other was then, this was what happened after you stop the hypoxia. And this buddy, the brown Norway, slows, and this sort of keeps its frequency up. So this is post-hypoxic frequency potentiation, and this is post-hypoxic decline. So each strain does a little bit different. And we could show that that was the quote, that's called the de Jour's phenomenon, if it goes up or down. In the brown Norway, it drops in frequency, it drops in minute ventilation, whereas the Sprague-Dawley goes up in both. And now I'm going to show you how does that relate to almost all the other traits we measured. But I'm going to do it in a fairly interesting way that I learned when I went to I did an internal sabbatical early aughts and I went to Milwaukee. So this is the correlation of post-hypoxic ventilation decline and de jour. So in the frequency, the change in ventilation between before and after the hypoxic was 0.8, brown Norway was 0.7, and the FT was 0.8. So it was preserved. That is any correlations. Frequency, not very much with frequency. There was no correlation. And this is ventilation. So this will be the correlation coding. So a 0.8 would be up here, and a 0 would be black. And then if you got down to this point, it would be the other side, 0.8, right? Now, the reason they do that is because we're very visual people. We understand sort of visual things. If I gave you a whole plate of numbers like this, which people do, you're not going to figure it out. How does de jour's correlate with other traits? So this is all the traits we did. This is oxygen consumption, CO2 production, RQ, frequency, all the way down. See, all these things. Here's the de jour's, right? The reoxygenation. And this is for the Sprague-Dawley. But you can see these correlations kind of cluster in certain areas, and they sort of make sense. But the de jour's here, down here, 1, 2, 3, going all the way up. Here's a little set with change in ventilation, frequency 5, change in ventilation in 5th frequency, that these things have a little bit of a difference. The rest is all around. The blacks are 0, right? And they go back and forth. But that's not the most important thing. Most important thing is, how is it different between the Sprague-Dawley and the brown Norway? And you see that it's all hooked up to. Now, both of these strains have babies, they're successful and some more than others. These are not, screwed up animals. These are just how animals use their genes to survive, and you can use them in different ways, and you can do those genes in different ways and attend them to different traits, and different traits will give you survival. Okay, so this is the first map that we made, which is we put unfortunately, this thing up here is terrible. Whatever this thing. What's the check? Okay, we got a credit. Get that out of there. Okay, welcome. Fine. So this is the rat genome. It doesn't have a sex, but it has, and this is what we found in terms of the reoxygenation the first minute, hypoxia the second minute. These are the different parts of the genome where various people are. And this is where other people had found things that might relate to sleep apnea, like POMC, which is an obesity gene. And this is hypercapnia, this is hypoxia, this is nitric oxide synthase 1, 2. Foster found something here. And you can see that this is a polygenic trait, and no one gene sort of contributes, and they contribute across genes. So I want to sort of go on a little bit here. This leptin, that's neat, O2, CO2, and leptin has face validity because people have looked at leptin and its control of breathing and putting it into the hypothalamus and things of that sort. All right, so let me see what I did here. Okay, so genetic variation produces differences in ventilatory response to hypoxia. CO2 linkage varies with exposure time. Several chromosomes are involved with at least a 6%. We could detect a 6% effects. Now, there may be 100 genes that have 1% or 2%, but you really want to look at things that have about a 6% effect. And the linkage is not to the same chromosomes with reoxygenation or with hypoxia and CO2. That was really fun. That as soon as the 5th minute of hypoxia showed the linkage, the first minute of reoxygenation, that was gone. That linkage was gone, completely gone. So, mouse studies, 2001 to present, a little different chamber, but the same thing. Sensors, vacuum sufficient. And here is the B6 and the AJ. This started because we were doing these rats, and we were sort of like not doing, and Feng Han came from Beijing, and he was working with, we said, well, you're gonna have to work, unfortunately, you're gonna have to work in the animal lab, because right at that time, our animal lab didn't have as much support as the human lab, and because he was a visitor, he couldn't work with the humans as much, so he's gonna work with the animals. And so he started working with the animals down there with Ted Dick, but they had mostly mice. So we said, well, let's do brown oryx and AJ strains. These are, again, 2 inbred strains. The B6 is the most common strain, which people knock in and knock out genes. The AJ is, it's a quirky little thing that they use a lot for pharmacology. I don't know why, but they use a lot for. Well, we're not the only ones that said that they breathe funny, and this is for Matthias, because his group showed that this B6 had apneas, which was just the pauses that kind of dropped out of nowhere, 2 or 3 breaths. And this was their recording that they had. And it was for a while, this was in 2008, we reported apneas and pauses 2002. And for 6 years, I'd be presenting at conferences, and everybody would look at me and say, well, has anybody replicated that? All right. That Kingman Strohl out there must have some magic to make those things do funny things, right? I mean, that's how you do it. You say, well, is that real? Is it because how you manipulated them, how you reoxygenated them, how you looked at them, whether you brought them up and you scared them to death, or you didn't scare them to death, whatever it is. So I showed that to say we weren't too crazy, 2006. And meanwhile, I'm trying to get funded for it, so that was hard. So what Feng Han found was this. So the air baseline, they breathe like that. Reoxygenation, the B6 slows. AJ keeps breathing pretty well. And the B6 started to have this sort of stuff. Now, this is a cartoon, but it goes on for about 30, 40 seconds and then goes away. But you obviously start something that creates an instability that is intermittent with apneas. And this is all central apneas. And people are saying, well, these are all central apneas. People have obstructive apneas. And I kept telling them that central apneas, you close the airway during central apneas and you start an obstructive apnea with decrease in drive. So recurrent apnea. So here you go, higher loop gain, right? That B6 has a over response and over suppression, and the AJ has a lower loop gain. And we found that even with size, we did a lot of things to sort of do that. A lot of publications came up. We did drug gene modification, acetazolamide. Now I have to show you what this is. This is the a Poincare plot in which the frequency of one breath is plotted against the frequency of another breath. And this is baseline before we give them anything. This is then hypoxia. Minute 1, minute 2, and reoxygenation. And what you can see in vehicle is you breathe a little more irregularly during that first minute. Then 5th minute, you settle in. And then post-hypoxia, you have all that irregular breathing. So it's really how big that dispersion is. Give acetazolamide, and the frequency is up a little bit, frequency is up a little bit, frequency is up a little bit, but you really have less dispersion, so we can treat. See, so we were all excited because that's acetazolamide for congestive heart failure with recurrent central apnea. That's pretty good. We did inhibition of dihydrosulfide, which is a drug you can use, and we did various drugs. We did inhibition of 7NI, in which we could prolong apneas of B6 without producing them in the AJ. So we can see there are now, whatever drugs we're going to use for sleep apnea, there's going to be a drug by gene interaction. I make it bet. Now, maybe that you have sleep apnea because you have a high loop gain, so you won't find it, but that's a proof of genetic transmission. We used this because it was here. One of the genetics people was making these things. The AJ is the donor, B6, and you breathe them for many generations, and you end up with mice that are homozygous for one chromosome. Now, so chromosome 2 is only from the AJ, and the rest of the genome is from the B6. Is that clear? You keep breeding them and breeding them and breeding them until you get there. And then you make them, and you can buy AJ, B6 AJs, you can buy a lot of chromosomal substitution strains now from Jackson Labs. So you just, they have the sperm there, so they just make it for you. So this is the response of negative is post-hypoxic frequency decline, positive short-term potentiation, and the AJ shows short-term potentiation. Before 7NI and after 7NI shows post-hypoxic frequency decline, whereas the B6 has post-hypoxic frequency decline, both of them. And then your recurrent apneas, RA, and this is one chromosome example from chromosome A1, A11, A14, A16. And what you can see is that the short-term potentiation varies now among strains, one way or the other. And then it's interesting, you have short-term potentiation, but this was the only strain here that had no recurrent apneas. So if you take chromosome 1 from the AJ and put it on the B6 background, it didn't have it anymore. All right, so what do we do with that? We cross them, right? We cross the B6 and the B6A1, you bring them down. You bring them down to the brother-sister mating, for instance, recombination, determine what it is. You find that the QTL is there. There was actually probably three parts of this correlation that are there, but we really focused on the QTL, but that was in which is this one here. These are identical on these, which is the ApoA2. So B6A1 chromosome, and then we had this region that we found in the animals in which they had this QTL, which didn't have any of these apneas, whereas the B6 does have apneas, the B6A1, and then you could take in the B6 alleles into this, and you would not have you would have apneas. So you could get them back and forth. And underneath this QTL gene expression array showed that ApoA2. Other ones were there, and we're finding out that means something. But the ApoA2 was there, and for sake of time, I'm going to kind of go through this pretty So you go apolipoprotein A2, what is it? Made in the liver, second most abundant carrier protein. It's also now made, we know it's made in the brain. Mouse and human associated with cholesterol levels and metabolism. 3 human cohorts report ApoA2 high fat diet, interaction with obesity, but no known role. We showed it had a role in the brain. We could show that ApoA2 is important and it was found in both the medulla and the pons. It doesn't relate to any of the drugs that we use to try to manipulate these things. And we went in, everybody said, well, it must be something wrong with the animal. We tested the mechanics of the lungs and chest airway. That was not it. We tested the nerves and muscles. They could do whatever they needed to do according to normal activity. We looked at their carotid body, and the carotid bodies were not particularly different. And so we're left here with this pons and medulla. We were fortunate that at the same time, the lab over on the 3rd floor was doing the perfused in situ preparation, phrenic nerve activity. This is then wild type. And this was the changes in the knockout. The knockout was much more regular. The wild type was irregular. That's the brown, that's the black 6. And that you can show that in a variety of statistical ways. That these things, here's a Poincaré plot of the fictive breathing of a brainstem, showing a lot of dispersion, and of a knockout that showed little dispersion. So where is it? Out of the prep is circulation time, lungs, carotid body to some extent. In the prep, though, is this brainstem reciprocal back and forth between NTS, pre-Betzinger complex, and the cochlear fusion. So I foolishly say that some of this instability is due to instability in that network, and I make an analogy to emergent ventricular arrhythmia. I don't know. So synaptic connections. These synaptic connections could be affected by ApoA2, either by its message regulating something else, or it could be because of the protein itself. We don't know. Weight gain on the low fat, on the high fat diet in knockouts is the same as the wild type. So it's similar, and yet there's no increase in post-hypoxic pauses when you make them obese. On the other hand, looks like we've got a gene that not only works on loop gain and instability, but also on body mass. We have a pleomorphic potential, and that's what I'm trying to look at. So where do we go from here? Well, ApoA2 is deterministic for pauses. It's not associated with weight, anxiety, sensation, rate of learning, or motor strength. Did not gain weight on a high-fat diet. And this is now, our region is over there on 1, which is a low CO2 interaction shown by another group, trying to create this map. And then we want to map this onto the human genome. So published work on strain differences in gene expression arrays and proteomics. I have that in the can. We'll see if that's good enough to publish. Ask the genetic experts to look for ApoA2 polymorphisms in these large data sets, interactions to BMI and to gain if they have it, and to PCRIT. I don't think there will be anything to PCRIT, maybe through BMI, at least BMI associated. And AHI. AHI is a hard one to link it to. We want to confirm where ApoA2 resides and create a pipeline to look at what its activities do. Collaborate with HDL cholesterol experts for brain function. There's still very little on that. Everybody's focused on Alzheimer's and ApoE and testing of chemosensory traits in humans according to genotype. So I would take their genotype, my hypothesis would be if you had a genotype of ApoE2 of a certain polymorphism, that you would respond to reoxygenation with respiratory instability. So I wanted to define breathing as a complex trait, explain how rodent models can be used to dissect gene actions, and describe the concept of a genetic map. A lot of people involved in this since 2001. Put Erico down there, but last name I should use it. We'll figure it out. Easy. All right. Okay. So that's the map of how I think of ventilatory behavior. It's a little different. It comes from the honeybee evolution group. The genetic, molecular, and cellular foundations makes hormones, nutrition, energy state, developmental programs, and growth, maturation, senescence, decision-making, that's handling, integration, reaction times, and hierarchy, that's neural system, and psychophysical constraints and information processing. Okay.